There.
And thank you for letting me present our work here. So in the first half of this presentation, I will show you how we use grid computing and workflow technology, and this was a pilot to check if we can use it for next generation sequencing. And in the second half of my presentation, I will show you how we applied this method for a larger scale study, so to see if we can scale this up. And I will end with some current challenges and a brief summary. So you might be familiar with pictures like this, and also from practice probably. So in this picture, in time, the capacity increase is depicted. One for Moore's law, that's this black line. So that's the increase in computing capacity in time. The red line is the actual CPU increase in capacity, which still follows this line. And the green bar here is the increase in capacity in sequencing. So you can already see here that we have computing problem and that we need to deal with it and by applying better algorithms to deal with the data. But we also need to invest in better IT infrastructures to deal with the data. What I liked about this picture is that also the increase in personnel is displayed, which is a straight line, which means that we need to work more efficient. So according to IT infrastructures, there are many options. One can buy a local cluster or set up a desktop grid, use a supercomputer or Hadoop or GPU cluster. One can use cloud computing, an international grid, maybe in the future DNA computing, and some of us might have access to national computing facilities. But, well, each system has its own benefits for certain algorithms, but the thing is that each system has its own interface and that we need to learn how all these systems work. So for our pilot, we decided to use grid computing. And the main reason is that it's all about sharing. So you share the resources, so computing and data storage resources, and it is an ideal platform to collaborate between different institutes. And it also uses open protocols. So in the Netherlands we have the Dutch grid, which is rolled out by the Big Grid project, and it is part of a larger international grid. And at the moment the most users are from high energy physics, but you see an increase of life scientists as well to use this grid. So in this project, several clusters and servers were installed at different universities and some companies and other institutes. And all these servers are connected via a very high-speed internet connection. So back to our institute. So it's organized like this. So for next generation sequencing, so we have a central sequencing facility with at the moment Roche 454 and a solid machine. And last week a new solid machine was installed. There's a central bioinformatics group who deals with this next generation sequencing data, and there are many research laboratories that want to do their experiments. So when they want to do their experiments, they go to the sequencing facility and also contact us. So I work here. And when we do the analysis, we use our own local server, or if we need to scale up, we use the grid now. But we don't use the grid directly with the interfaces that are in there, but we use it via services that were developed by the eBioscience team, but I will come to that later. So we are closely collaborating with this group. So I will give you an example. So this was the pilot project. It was in collaboration with the Virus Discovery Unit. So this is Michel de Vries and Lia van der Hoek. And they have developed a laboratory method to extract viral RNA and DNA from samples. And they sequence their fragments on the ROS454, so they have many experiments already. And the goal is to identify known and discover new viruses in these samples. So what they want to do is to blast these sequences against the GenBank database. But since many new viruses are deposited to the GenBank database, they also want to repeat this analysis after each GenBank release. And want to repeat it on all their earlier experiments as well. So for this purpose, we built a workflow which looks like this. There's a conversion step from the ROS format, SFF to FASTA. And in the second step, these sequences are aligned against GenBank. So how we implemented this is depicted here. So you start off with an executable or script. It can be any executable or script that can be run from the command line. And then we describe it as a so-called workflow component. So you make an XML description, and for those familiar with Galaxy, it's a bit like this tool description that you have there. So you define what you want to execute, and you define what the in and output data is. We can do the same for BLAST. Here we have two different inputs. So you have the sequence data that you want to align and one or more databases. And BLAST result will come out. Now, when you have more than one input, you can specify how you want to combine it. So you can choose, for instance, between cross product or dot product. So cross is comparing everything against everything, and dot product is comparing pairwise. In this case, we want to compare all the earlier experiments with several databases, so that's a cross product. And then we can connect all these components in a workflow, which is basically also an XML description, and where you state how these components are connected. So the output from this component will go into this BLAST component. So now that we have a description, we want to run it on the grid. And this is an architecture that was developed by the eBioscience team. So it was built on top of the services that were provided by the BigWit project. And the most important part is this workflow service. That was developed in France. It's called Moteur. And it's using Diam as a pilot framework. And when you give your input sequences and reference databases or other inputs, this workflow service will sort out what combinations need to be made to run parallel on the grid. So it will automatically generate the job description, so to say, and submit those jobs. When they are running, the jobs are monitored via monitoring service. But the bioinformaticians or the biomedical end users, they will generally not see these layers, but they will only see the top layer. So that's the user interface. So there are three right now. There's a command line interface. There's something called the V-browser, which I will show in the next slide. And if you have a production workflow, then you can use it from a web portal as well. So the V-browser, that's one of the interfaces. It looks like this. So it's an Explorer-like interface. And you can access grid resources, so grid storage, but you can also access local servers or your own PC. And you can drag and drop files from one resource to the other. The other characteristic of this browser is that you can use plugins. So one of the plugins is for this Moteur workflow service. So if you choose a particular workflow that was already built by one of the bioinformaticians, this plugin will pop up, and then you can insert here one or more sequences or experiments that you want to compare, and then click on Run Workflow. So what you will see then is a result of this monitoring service. So you will see at what the status is of each jobs. So how many jobs are running, how many have failed, and so on. So As a result, for the speed up for this pilot project, we have compared 15 experiments at once against 2 sections of the GenBank database. So ribosomal sequences and the viral section of GenBank. The total CPU time was about 17 days. But the workflow runtime, because these were all processed in parallel, was about 14 hours. So it's about a 30 times speed up. And the other thing that we thought was nice as an experience in this project is that we really like the workflow strategy. It allows for agile development. You can easily describe command line programs in an XML description. You can reuse components if you want your pipeline or workflow slightly different. And you don't need to program anything extra if you want to iterate over your files. So that also saves a lot of time. And maybe a nice side thing is that you capture your knowledge about a particular experiment or analysis steps in this workflow visually. So at the moment we have implemented workflow components for several programs already, so BWA, SumTools, VARScan, ANOVA, you name it. And we use that a lot daily now for our in-house experiments, especially the Roche experiment, especially the solid experiments. But at the end of last year, we got involved in a even larger project. It's called the Genome of the Netherlands project. And in this project, 250 trios, so father, mother, son, are sequenced. The entire whole genome is sequenced. And the goal is to enrich biobanks, and it will provide a reference set for disease studies. So this sequencing data, it consists of 770 samples, so a little bit more than the 250 trios. And it's about 45 terabytes of data, which comes in on a hard drive and needs to be put on some server or grid first. There are many partners involved, mostly nationally, but also a few foreign partners. And well, if you want to know more about this project, you can go to these 2 websites. The analysis at the moment is done on distributed sites. So a large part of the analysis is done on a very big cluster here in Groningen. And there are also some local sites that do parts of the analysis. And there are 2 groups that are using the grid now for part of the data. So that's what I would like to show here. So we decided that there is a standard pipeline that we want to use on all the sites that are calculating on this data. It's a pipeline that's similar to what is used at the Broad Institute, and it is implemented by Freek van Dijk from Groningen. And we ported this in this workflow methodology to see how that goes on the grid. So it consists of a BWA alignment and then some steps to fix the alignment. So in the end you will have a clean alignment file. So at the moment 160 samples are analyzed on the Dutch grid. Development started at the end of last year for this part. And the main part of the calculation for these samples were done since the end of March. So the total CPU time here was 5.5 years, and so that's in March, in 4 months. And this space used on the worker nodes was about 300 terabytes. And that already sounds nice, but still you need to make improvements to make it even faster. So there are a lot of challenges when you work on these type of projects and this kind of data. One of the things is that many things can go wrong along the way, so many jobs can fail, and you don't want to go through each error log from each individual job, so this error handling needs to be more generalized so that you exactly know, okay, what went wrong and how many times did it went wrong. So work is needed there. There's also work needed for data management. So the grid works as follows. So you submit a job to a particular worker node, then it will fetch all the data, do the analysis, and then copy the data back to storage. So there's a lot of redundant data transfer, and preferably you would like to have some kind of caching system, or just bring your job to a location where the data actually is. So there is also a lot of work that needs to be done. Very important issue is the data protection, especially if you would like to use these type of systems for more sensitive data. Then you can think of encryption of the data or other solutions. And for bioinformatics in general, it's important to track the provenance, which means that if you have your result file, that you know exactly how you came to that result. So what kind of program did you use, what kind of version, what were the parameters, and so on. And what we also would like to have is a transparent addition of other resources. So preferably not by changing the workflow, but by changing the underlying layer, so that you can add local clusters or maybe even cloud services. So to summarize, some more research and development is needed in this e-bioscience field. Part of it for these reasons, but also to provide easier interfaces to those IT infrastructures. I think the latest IT infrastructures are really needed for the analysis of next generation sequencing. And workflow technology can help to quickly implement bioinformatics software on different infrastructures. So it's separate, really. So you have two topics, the workflow development and the infrastructure. So there are a lot of people involved in this type of work, and I can only name a few. So the Genome of the Netherlands project, that is led by Siska Wijminga from Groningen, and Morris Swartz is leading the bioinformatics part. The work on the pilot project was together with the Virus Discovery Unit, so Lia van der Hoek and Michel de Vries. And the sequencing was done at the lab from Frank Baas. I would like to thank the BigGrid project. They provide grid resources, but also the support. And the workflow system and language was developed in France. So Johan Montouillat and Tristan Clatar. And I work here at the Bioinformatics Laboratory that is headed by Antoine van Kampen. And a part of it is the e-bioscience team who's working on the infrastructure, which is led by Silvia Olabarriaga. So I'd like to thank you for your attention and take some questions if there's time.